We'll start with some basic introduction. So today is the online session about using the EBRAINS interactive viewer to analyze the brain. And this session today will be given by Kimberley, Lothmann, Thönnißen, and me. And we will split the today's session in some parts. So I will start with a basic introduction. Of course, this will be a bit redundant with the talks from yesterday, but I think we cannot assume that everybody was there yesterday, and we will try to keep it short. The second part is really a live demo about the Zebra Explorer given by Kimberley, and then we will also show the tool Voluba, which is used to spatially anchor volumes of interest in the Jülich Brain Atlas, or in the multi-level brain atlas. And afterwards, we have a hands-on session when we prepared some little exercises, some ideas to start playing with the viewer to get a feeling for the use of the Zebra Explorer. And at the end, we will also wrap up the hands-on session, try to answer questions, and go through those little exercises, questions we wrote down in our document. So as I said, this might be a bit redundant, so I will be fast. The atlas concept consists of several steps, something like a hierarchy. So we have an atlas, which if we take the example from yesterday and compare it to classical maps, we have the different species which are included, like human, rat, mouse, even a monkey atlas, which is not listed here. You can compare it to different planets, for example, if you want to stay in the normal example of atlases. Then we have different reference bases. In our case here, especially for the human atlas, it's the MNI reference base, which is a typically used low resolution reference base with 1 millimeter isotropic resolution. The big brain, which is a higher resolution reference base, and FS average, which is a surface-based reference base. We can compare it to different geographic coordinate systems. It's a coordinate space for the for points, maps, or volumes of interest within an atlas. Then we have different parcellations. Within our talk in the live demo, we'll mainly focus on the Jülich Brain Atlas, which is one parcellation scheme which is included for the human atlas. This is comparable to different countries, continents, time zones, so different kinds of parcellation you apply to a reference space of an atlas. And the last point in the hierarchy are those regional features. For example, if you have receptor densities, cell densities, functional data for different areas. And if you stay in the example of a classical map, this is something if you go to Google Maps and see for the opening hours of a supermarket next to you, something like this, so more precise information about a specific region or point of interest. So as I said, we have different atlases at the moment, and we will see this in live in a few minutes. We have the human atlas, which is typically used by us, but there's also the Waxworm Space Atlas, Mouse Atlas from Allen Brain Institute, and also Monkey Atlas. So this is the first level of hierarchy. And I'm always pointing here to this wording of atlas parcellation and reference scheme, because this is something which is important, especially for the Python part tomorrow. Because every time you access some data using the Python library, you have to define which kind of atlas, which kind of reference space, which kind of parcellation. Therefore, I try to keep the wording here consistent so that it makes it easier for the follow-up steps. The next point of the hierarchy are the reference spaces. One of the typically used one is the MNI 152 reference space, which is included. This is a reference space built of the measurement of 152 and not patients, donors, and they build one mean reference brain, which is quite often used in the community. It's a very common template with 1 millimeter isotropic resolution. As Katrin and Timo yesterday mentioned, there's a trend of higher resolution because if you analyze something in a volume of interest with nanometer scale or micrometer scale, it does not make sense to anchor this one in 1 millimeter reference space because it will simply be lost. So therefore, you also have to increase the resolution of the reference space. which was done for the big brain, which is one of the template which is also included in the atlas. It was built out of 7,400 sections, which were scanned with 20 micrometer resolution in plane and reconstructed to one model. And yeah, you can switch between those templates. And this is something which is really important, and this is a typical problem we have in neuroscience. If we want to compare different modalities or different data sources, we have to use reference spaces, because each brain, as we heard yesterday, and of course all of you know, each brain is completely individual. And if we want to compare results from individual brains or from different sources, we have to choose a common reference space to bring all the data sets in one common space. And this is something which is internally solved by the Zebra Explorer, and also, of course, by Zebra Python, because everything we see later on in the viewer is based on Zebra Python. So everything we can visualize and see in the Explorer can also be extracted programmatically if you use the Python library. And there's also transformations are included, so that you really can link the different scales from the microscopic level of the big brain up to classical reference spaces like MNI or Colin, or even jump to the surface world of the FS average surface. The next step of the hierarchy after the atlas and the reference space is the parcellation. Here I will mainly focus on the Jülich brain, because this is something which is provided here by our institute from the group of Cartoon-Almonds. And those parcellations of the Jülich brain atlas are based on histological sections. We have heard about those sections a lot yesterday. So again, we cut the brain in about 7,000 sections. Of course, this depends on the cutting direction. Most of them were cut in the frontal direction. Then they are stained for cell bodies, and we have a section thickness of about 20 micrometer, which is about half of the thickness of a human hair. So they are really thin. And then we are looking for structural differences. We have already seen this very easy example yesterday, the border between the primary and the secondary visual cortex. Primary cortex on the right side, secondary visual cortex on the left side. And it's quite obvious that something changes in the structure. But it's not always that obvious. So if we are coming to higher associative areas, we need some quantification to really define borders between adjacent areas. And this is be done by creating line profiles along the cortical ribbon. And here we can see two example profiles from neighboring areas. And if we look closely, they look slightly different. But of course, there's always a lot of noise in human tissue. So if we really work with real tissue, we have little artifacts, blood vessels, something like this. So therefore, we have to extract a lot of those profiles and work with group measurements or blocks of profiles and compare the shape of those blocks of profiles against each other. This is done by moving along the cortical rim, and then we compare different groups and apply some statistical tests. And if we find significant maxima, then we can acquire this one as a real scientific border, as a real border between adjacent areas. Of course, this have to be verified in the tissue itself, but at least it's a very strong hint. And we have to reproduce those maxima and following sections to be really sure that there's a structural border which is significant and real. The second part for the parcellation is that those brains do all look different. So every brain is different, like a fingerprint. And here you can see it for the primary visual cortex, the red area, which is really very easy to see. So you can be absolutely sure about those borders, but there's really a huge variation between the individual brains. And that's why the Jülich brain parcellation provides two kinds of maps. So we analyze 10 individual brains, bring them to the same reference space, superimpose all those 10 results, and create a probabilistic map. So red here means that the specific area was found in 10 out of 10 brains with a lower probability in the periphery of the area. And if we would combine those probabilistic maps, we would create a huge overlap because of this variability of the individual brains. Therefore, that's why there's an additional map provided, which is the maximum probability map. The difference here is that overlapping maps, the voxel which have overlapping maps are assigned to this area with the highest probability. So it's a kind of winner takes it all model. By doing this, we can create sharp borders between the adjacent areas. But of course, we lose a bit of the probabilistic information. So whenever it's possible, we prefer to work with the probabilistic maps because they provide the highest degree of information. But of course, the maximum probability map is a kind of aggregated map, also provides probabilistic information, but in a simplified way. It's not only the Jülich Brain Atlas, and Kimberley will show it to you in more detail in a few minutes. So we have the Jülich Brain Atlas with its different reference spaces, but there are also parcellations for fiber bundles or of functional nodes coming from different labs of the EBRAINS infrastructure, and they are all merged and brought together in this huge Atlas project. And again, here we can link those different areas. So it's not only the pure reference spaces and the coordinates which are linked, so we also have corresponding regions. And it really depends if those areas were mapped in all reference spaces. So between colon and MNI is a simple transformation. Sometimes those maps are also available in the native big brain space. It depends, but whenever it's possible, they are directly connected, and we can really switch between the reference spaces and choose the one which is best used for us. And the last point of the hierarchy are those regional features. And here we are really completely open. It depends on the data sources which are available for the specific areas. So for some areas, we have receptor densities. For some areas, we have PLI data. We have just prepared a very large data set with laminar-specific cell counts for more than 100 areas of the Jülich Brain Atlas, which will also be introduced using the Atlas. But this really depends on what's there for the specific area. And here the system is also really open. So if somebody has a specific volume of interest, which can be spatially or semantically linked to a specific area, it can be brought into the Atlas after some step of creation, which will be shown by Liuba on the last day of this course here. One point which is extremely important are those yellow screenshots here on the right side, because every data set you see, even every map, every reference space is deeply curated within the EBRAINS Knowledge Graph. So it's not only a label which was created by someone, so there are really a lot of metadata information which makes it reproducible. So whenever you select one region, you can see the specific scientific publication. You can download the data, you can see whom to cite if you use the data set, and all those things are more or less machine readable. So it really, there's a huge data source which can be used by everyone who wants to analyze those data more programmatically, but you can also directly see it through the user, through the viewer, which will be shown by Kimberley. So at the end, we have the complete atlas, which consists of those different hierarchical level. So we have different spatial resolutions. We have different maps or parcellation schemes. We have multimodal features, and all this together creates the atlas as a tool, which can be accessed through a web page, but also through this Python library, which will be shown by Timo tomorrow. One last thing for the atlas viewer, it's also open in the sense that you can bring your own plugins to the Zebra Explorer, which was done here for this UJEX tool. Katrin and Timo also featured it yesterday. So this is a real multimodal combination of different labs all over the world. The upper row shows tissue samples which were extracted by the Allen Brain Institute, and they also provided the MNI coordinates for those tissue samples. And since we are working with the same reference space, we can use our probabilistic maps to extract specific tissue samples from all Allen Brain donors and to analyze them with different statistical tests to look for differences of the gene expressions of genes of interest. So here it's a complete loop as an example. So in the upper left frame, you can see the cytoarchitecture. This was the starting point to really create those maps. then we applied those maps and the borders measured on 10 different brains to the template space. And by doing so, we have created the probabilistic maps. Those maps can be used to functionally decode the area and look for different functional connectivity, also possible using the Zebra Explorer. And this was done here for this specific example, and we found that there are region-specific differences between the two areas covering the frontal pole. And those areas, and even so the connections and the functions, indicate that there might be different behavior in terms of major depression disorder. And therefore, we have used those analyses and compared the maps with cohorts of depressed patient and control patients. And there we also found volume-specific differences between the two groups. And so we were quite sure that there's something happening in major depression disorder in those specific areas. And then as a last step, we used UJEX to extract the specific tissue samples from Allen brain and applied a candidate gene list also from a list from Allen brain, and looked for differences between the tissue samples located in area FP1, it's the frontal pole, and FP2, which is the medial part of the frontal pole. And indeed, there were also significant differences for some genes of those two areas. So you can really build a cascade of analysis built on those maps, starting from cytoarchitecture up to differential gene expression between areas. And this is how it looks like in the viewer, but this can best be seen in the live view, and Kimberley will do it in a few seconds. Just one teaser here. The Zebra Explorer is one very basic and intuitive and well-looking starting point for all the data sets we have shown and will show in the future. But there's also Zebra Python running behind the scenes. So everything we do can also be done with Zebra Python. And it's very obvious here for this UJEX, because you can do a starting analysis, but of course you cannot create a study for publication in an online viewer. So therefore you can only do it here with very limited statistical power. But if you want to do a real analysis and do this by a Python notebook, you can interactively press a button. This will open in a Python notebook, which is completely configured by the selection you made in the Zebra Explorer. And then you can really start the real analysis and produce real-world data to publish your data using different parcellation schemes, not only with the Jülich brain, so for all the different parcellation schemes which are available. Yeah, and by having this said, I would like to hand over to Kim to start with a live demo of the Zebra Explorer.
Hello, everyone. Thank you, Sebastian, for the nice introduction. First of all, I will send you the link to Zebra Explorer in the chat. And if you like, you can do simultaneously what I'm doing and test if everything runs for you. And after that, we have a little exercise where you can also test everything that you want to see. So first of all, Zebra Explorer is a tool with a lot of stuff, as you already know, and I will start to show you which atlases we have. So what you can see here, this is on your starting page, is the multi-level human brain atlas. So when you open the atlas, you see on the ground here, we have the atlas, we have template and parcellation. And if we have a look at the atlas, I want to show you the different atlases of different species that we have here. For example, what most of the people worked with in my PhD was the Allen Brain Mouse Atlas. And what I worked with was a PDF with every section of a mouse brain inside. And this was kind of horrible to work with. And now we have it in a 3D reconstruction. We have sagittal view, we have horizontal view, and the coronal view. And here you can see, if you go over the areas, you see which area you have and in which layer you are. So this makes it a lot easier to work with your mouse data if you have this atlas here. You can scroll through it from every direction as you wish, and use this template to work with your data here, for example. What we also have is the Waxholm Red Atlas. I see there are a lot of people now on the Atlas. That's nice because it takes a little bit more time to load at this moment. So the Red Atlas is a little bit more huge, so it takes a little while to build the 3D reconstruction here on the right side. And it's the same principle as the other atlases. We have our 4 views here, and you can scroll through the atlas. You see, you can click on an area and there are information opening. We have features to every area. Everything that is inside this atlas, you can see in Zebra Explorer. And if we switch to the last one, because I just want to work a little bit more with the human atlas, I just want to show you what we also have in our EBRAINS atlas, that we have different species, is the monkey atlas. This atlas is based on macaque. And if you have any questions, you will always find this info button down here and click on it, and you can see all the details that you have to know. So in this case, we have the MeBrains multi-level macaque brain atlas. I will show you this point later. And now we go back to our human brain atlas. So this is what we see when we open it. And there are different tools that we have in this one tool. So let's start. For me, at the beginning, I am a neuroscientist. So what I did was creating one of these areas. So I made the cytoarchitectonic mapping on it. And what we get at the end are coordinates. And friends of mine working with MRI. So what they have at the end are coordinates. So I have an area and don't know the function, and they have functions and don't know the area. So what we implemented in this atlas is that you can search for an area by coordinates. This is quite easy. If you see the coordinates up here, this is based on this cursor symbol here, the red and blue lines. This is the point of this point here is where you are actually located. And if we now press this pencil button here, there's a window opening where you can put in your MNI coordinate. Just as an example, I will enter some coordinates to show you the options now, because we can, for example, navigate to the point if we just want to have a look, where is this area located, and Zebra Explorer switches directly to our coordinates. Now we see our cursor on this area, and if we go with our mouse over it without clicking, we just see it's area 45. So we are talking about Lothmann here. What we can also do is giving our coordinates. They are now inside because I saved it, and select this point. Then there are some map values opening, as you can see here on the right side, and he tells us area 45 is the area that is most common here, because we have probabilistic maps. The maps can overlap. So the unprobabilistic map space overlaps here, for example, for area 44. And as we can see here on the right side in the sagittal view, now area 44 is the next area that we have. So that's why this is the next area that will be here in the list. And another areas where the statistic tells us it is not common that this area will be here, but it's nearby. So if you have an area where you a coordinate where you don't know which area it is, you can find here the most probable area. If we go back, we see again that this view is being held, but we have different options. So this was now finding an area by coordinate. What we can also do, sometimes we have an area, we want to see it, then we can search for a region here. If we search for a region, we can type in, for example, area 4. Now he tells us, what do you want? 44, 45, left, right, area 4P. Let's take, because I'm down here now, area 4A. You can just simply click on it, and you see this bar opens on the left side. Here you now have all the features that we have in Zebra Explorer. So for example, at the moment, we are in the cytoarchitectonic map V3. This is the actual version. Next one will come soon, so stay tuned. I will give you a link after my talk that you are able to stay tuned. And what we can now do here is going to the region centroid. So no matter where your cursor is at the moment, you can directly go and switch to our probabilistic map to the region centroid. If you want to have a closer look, you can press this little brain up here. So the region is now still active, but you can have a closer look to it. You can scroll through it. You can do whatever you want and come back to what you have searched here. Then, if you're interested in this region, for example, now here, area 4A, you can directly go to the, as Sebastian also mentioned, the knowledge graph. If we click on this link, you get a direct connection to the probabilistic cytoarchitectonic map of the area that you are at the moment. This is not everything that you can find here, because here is now the actual paper to this area that you can cite. But if you cite it, you can click on how to cite, and then you will always also get our science paper for the whole atlas. So the Jülich Brain Atlas paper. And you can see also here that we don't only have the paper to this area, we also have the data set. So if you want to use a probabilistic map of an area, you can always cite the data set with this link here. You can also get the data. If you click on Get Data, you find everything to download here. We find also more publications. These are the same that we had to add how to cite here, the specimens, and the how to use. So everything you have to know is in the knowledge graph. And in the overview, you can also see the project here is the Jülich Brain Cytoarchitectonic Atlas. And this is necessary if you use data that is linked to our atlas, for example, PLI data, something else, not cytoarchitectonic projects, you will see it here. Okay, so let's go back. What we also have here are different features. If we click on that, we see that you find here on cellular level the intensity profiles based on the big brain, and we will switch to the big brain soon. And you can scroll down and find a lot here. But what we also have are the Thönnißen Atlas data that is linked to our Jülich Brain Atlas on this side. The connectivity, I will go, oh, let's do it now. We have it opened now, so let's talk about that. Well, because what you can also see if you have an active area at the moment, you can see the connectivity of this area. We have here different modalities. At the moment, we have the functional connectivity selected. You can have a look at the streamline counts, the streamline length, or the tracing connectivity. If we now choose the streamline counts, you see that the atlas switches in view. You can also select the cohort. So now it's the HCP. You can also go on the 1,000 brain studies, but at the moment we have some service under maintenance. So we have a little stuff that is not working quite good at the moment, but it will be in the list the next time. And at the moment we have the subject selected. What you can also do to have just an overview on what is going on here in the streamline counts, you can select the average or the different view here. Everything that you are doing in Zebra Explorer, you can export, because you can just get the data on your notebook to work with it in Zebra Python, for example, or just making your own diagrams after that. So if you want to have this data, you can click on export, and then he will ask you if you want the regional or the data set. If you have questions, there's always the information button that you can press, and then you find more information to that. So, but, oops. As I told you, we had now a look on the different atlases, but what we also have, for example, now for the human brain atlas, we have different templates. Where we were now is the ICBM template. What we also have is the MNI column. Here you will also see that the 3D reconstruction looks a little bit different. I also prefer this for publications, for example. And if you now find something interesting here, because the content is the same, it's just another template, and you want to have a picture of something interesting, you can find the screenshot tool up here. So you can make a screenshot for whatever you want here of your area of interest. There are a few other templates, as Sebastian mentioned, for example, the FS average surface tool. Here, the thing is, for example, I work with some areas that are inside of a sulcus. If you have an area inside your sulcus, you can use also the coronal view. Okay, that's possible. But if you want to just have an overview on where am I in the human brain, it's quite good to have this kind of view, because you can not only zoom in, you can also show the inflated view. And if we use that, we can see, for example, this area, area 4, area 3, and area 3B here that are really hard to see because they're in a sulcus. So for this case, you can use the inflated view of the FS average. And for some other cases, we can also get to the white matter view and see our areas in the sulcus, for example. And also here you can turn off no, that's this one, the grid, and take photos for your poster or publication. The last one, I want to do it a little bit different, because now I want to switch to the big brain with you. The big brain, the special thing about this is, as you already know, we have here the high resolution pictures. So if we have high resolution sections of the brain, we can also scroll through it, but since our servers are under maintenance, I can't show you this interactively right now, but I will show you a little video for that, but not at the moment. But if you want to have a look at your area of interest in the big brain, you can go to Zebra Explorer. I will start at the beginning where you all start. You can search for the region that you are interested in. Let's take again area, we had 4A. On the left side, you can click on it. We know this now. We go to the region centroid, click on it again, and now you can just switch the template. So the view that you have at the moment, we are now at the centroid of area 4P, will stay if you switch the template. So if we now switch to the big brain template, which is a single brain, it will hold your view. So not only your view, it will go to the registered area 4P in the big brain. This takes a little while. There it is. So now we just wait a little bit more. Now it is here. So we see also, if I zoom in, the cursor is at the same position as in MNI space. So if you want to work with your area of interest that you know from MNI space, you can easily here switch to the high resolution big brain. What we see now is not the high resolution. This is now, as you see, a little bit blurry, but unfortunately we can't switch at the moment to the 1 micron scan. But even in this view, you can see that we have here bigger cells, because we are in area 4P and see BETS cells here. And the special thing is also here that we have the reconstruction in a sagittal view, where you also can see the bigger BETS cells, and here in the horizontal view. And now talking about BETS cells, most of the time, or every time, BETS cells are in layer 5. So when we see this here now, as a scientist, I know where layer 5 is, but sometimes some regions are a little bit different. So if we now want to see where is which layer, we have a project of Konrad Wachstel implemented in Zebra Explorer. And this is the time where I tell you something also about the parcellation here, because what we can choose is cortical layers for the big brain. If I click that, it opens again. It takes a little while. And now you see the view stays. And here are our bed cells in layer 5. So this helps sometimes to have an overview on the 6 layers of the cortex. This is just for 6 layers. So if you have a layer 3, for example, that has a layer 3, A, B, and C, you can't see it here. But for the first overview of a new area that you're interested in, it helps. So I'm running out of time a little bit, but I have a few things to show. So this was the big brain now. Since we don't have the pictures, I will show you a short video. After this video, I need a few more minutes to show a little bit more about the atlas, because we have really a lot of stuff to show you. So I will now show you the video of the high resolution sections. So this is our website. This is what I showed you. So now if you have the section opened, you can just choose the corona view. I took this just because I want to show you the high resolution sections quite big. And now you can search for an anchored section. This is one possibility that you have. If you click on it, the high resolution image will pop up and you can zoom into cellular level. So you see here the giant cells. I'm sorry, because this is a video playing now. What you can also, there's a different kind of usage. You can scroll through the big brain to the 1 micron sections. That's one possibility, and you stop where you want to see something. Or what you can also do is have an anchored section to your current view. That's what you can see here. I will stop that, because if you have one section selected, or for example, one area, you can go to spatial features, and then you see these little yellow lines. As I said, these servers are under maintenance, so at the moment this is not possible. And every of this yellow line is one micron section. Then you can just simply click on one yellow line and the 1 micron section will pop up. So it opens in the same window. The same thing with the cortical layers works also if you have the 1 micron section opened. This means here, when you search for a region of interest or you scroll through the brain, you can see the cortical layer. Turn it to full screen. And now you see there's not the 1 micron resolution underneath this layer. But what you can also do is going to the spatial feature and choose the selected 1 micron view for this section. Then he will switch to the high resolution section and you go back to the region of your interest and have a look at the layers then. Because what you can do with the cortical layers is switching the opacity of your 1 micron section so you can see the layers. So now you can see in your 1 micron view where are which layers. So now I will stop the video again and go back to Zebra Explorer. Because what we didn't talk about, and I know it's a lot of stuff that I talk about because we have a lot of stuff in our atlas implemented. What we can also do is taking another parcellation. As Sebastian mentioned, we have the fiber architecture in M&I space here. This is a project of Jeff Mangin from France. So here we now selected the deep fiber bundles. What we also have here is the superficial fiber bundles, or you can also directly go to the whole project and see everything here. This is the HCP project. As I mentioned, if you have any questions, go to the information, and there you can see all the information to now the Human Connectome project, MRI dataset. So this is linked to every project that we have implemented. Another parcellation that I wanted to show you is a functional mode. This is from Bertrand Thiriot, the Daifumo Atlas. And what you can select here, this is depending on you, which selection do you want. The functional modes has 1024 areas. And then it switches, and it's a little bit different now because here you have the component that you can search for your area. For example, if we go to area 4, you can directly switch to the functional modes parcellation, look where your cursor is, then you find the component that you are interested in and enter this in the Python script of the Daifumo Atlas to get functional modes. So information about the function. I know it's a lot of stuff, but last but not least, since also this was mentioned, I wanted to show you JUGEX. For JUGEX, we have the implemented tool in Zebra Explorer in a small version. So if you just want to have a short overview of everything, you can go here to JUGEX, where you can also take a screenshot. This is a plugin tool. And here you can select a region of interest. For example, V1 inside this first region of interest, and FP1, that's a very nice area here. And I just take the whole area and not in left or right. You can just do that too. And here you can select a gene. I just take the first one that pops up here. You can switch in permutations. I will leave it at 100 because it takes a while. The threshold is at 0.2. And now I can click on Run, or I can click on Notebook. If I click on Run, you will see the result in a minute. If I click on Notebook, it switches, so you can use everything that you selected here in your Zebra Python notebook. So as Sebastian showed, it will pop up in Python, and you can use the code there with everything that you selected in Zebra Explorer. So it will be the same information. Okay, unfortunately, there is too much going on at the moment, so I just quitted the operation, and this is what he tells me. The operation now failed. We can do this the next time when not so many people are on this server at the moment. Okay, so this is everything in a nutshell to Zebra Explorer. It is a lot. I guess it will also be a lot more if I have more time now. But as I told you, we have a new version every time that we make a new area, for example, that we have a new cytoarchitectonic mapping. If you want to stay tuned, I will post you our LinkedIn for the Jülich Brain Atlas. And I would be very happy if you follow this page, because everything that is new, I will put it on this side. So you have an overview if we have new maps, if there's a new gap map that is closed, I will post everything here. So follow us on LinkedIn, and thank you for your attention. If you have any question, feel free to ask now or later in the hands-on session. Thank you. I will now give the voice to Julia.
Thank you. Yeah, I think we will have a look at the chat and answer questions there if there is something coming up, because I need to speed up a bit now. Thank you. Okay. Okay, so what I want to show you, let me close these windows, is Voluba. So I will try now to switch between the presentation and the browser. Okay, that seems to work. So as Sebastian already explained, we have a tool called Voluba, where you can anchor high-resolution volumes of interest to our atlases that Kim just showed. So for example, the 1 micron sections were also anchored with Voluba. So first of all, I want to give a little motivation why we want to do this kind of anchoring, because if you have a partial volume, for example, with high resolution, usually people will not be able to tell where it was extracted from, only you know it. So if you want to share it with other people, they might ask questions about where you extracted the tissue from, what its orientation is, and maybe you yourself might also be interested in regions that are inside of your tissue, which are overlapping, or if there are other data sets that might be interesting for your research. So now if we anchor our tissue to the big brain, for example, we can directly get some information out of that by only looking at it in the viewer. So for example, we can directly see that this is tissue from the hippocampus. We already know the orientations because we know we have here the coronal, sagittal, and horizontal view. If we scroll through the atlas, we can also select all the regions that Kim showed and all the data set linked to it. I will do that later hands-on, so then you can also see how you can do that with your own data set. Just in general, there are a few tools in eBRAINS that you can use to anchor image data in general to the atlases, but we are focusing on Voluba today, which is especially optimized for high resolution, large partial volumes. Okay, so I want to paste now some links into the chat so that you can follow around if you want, but there will also be an exercise later if you don't want to join now. And you can also have a look at the documentation where there is also a tutorial, a step-by-step tutorial, which is exactly the same that we're doing now. So if you want to do it later again, just have a look there. Okay, so a workflow in Voluba starts with selecting reference space and input volume. So as reference volumes, we have the ones that Kim already showed. So the rat, the mouse, and the human brain. And then afterwards, you also select the incoming volume. So now I switch to my browser and show you how that looks in live. So for now, we select the big brain, so the human template. And as an example today, I want to anchor the hippocampus. If you have own data, you can also log in with either eBRAINS or OrgID, and then you can also upload your own data, which needs to be in Nifty format. And then you can also have a look at your own data and anchor it to the big brain. But for now, I take a publicly available volume. And then we click on Start. And the handling is the same as in Zebra Explorer. So you can zoom with your mouse wheel. And now we switch back to the presentation because the next step is adjusting the appearance of the input image. As you already saw, we have, for example, background in our image that we are not interested in, or if it's a grayscale image, maybe we cannot really see the structures that we are interested in for anchoring. So what we can do in Voluba now is we click on Volume Filter. And there we can adjust the settings. So for example, we can hide everything that is black, so all the background, or we can also select a color map. For example, I just take Viridis for now so that we can see the structures a bit better. So you can drag and drop the volume around just as you like. And you can also adjust the opacity so that you can see the volume better if you want to focus on a specific part of your data. Now, the next step that we are going to do is really doing a transformation. So as you already saw now, maybe if you know how the hippocampus looks like, the views were not aligned. So let me switch back again. We can see over here, let me just move that a bit. that this is the coronal view of a hippocampus, but in here, let me zoom again, we have the horizontal view. So this doesn't match, which means we need to do a transformation. You can do transformations by clicking on the brain icon, and then a dialog pops up where you can see all the transformations that can be applied. For example, we can scale the image down, which makes sense now because we can see that it is quite big. Yeah. So you can also use the slider, which makes it a bit easier if you have a bigger volume. And then you can also do translation, which is just moving the image, but you can also do that with the mouse as I am just doing now. Okay, let's do it a bit smaller. And if you have the feeling you're losing the volume, you just need to move it inside this colored lines, then you will get track of it again. Okay, and now we can do, for example, a rotation. You can type in values over here. You can use the sliders, or if you want to do a rotation by 90 degrees, you can also just use the buttons up here. So for example, we can rotate it on the x-axis, which makes it, I can show it here side by side, a bit more reasonable than before. So this is only a rough anchoring. If you want to do now a very precise anchoring, you can also do a refinement with landmarks. And based on these landmarks, Voluba can then calculate a transformation, which is linear. So we are not doing non-linear transformation, which would do deformations of the volume. This only moves the volume, rotates the volume, or scales it. So it does not alter the morphology of your tissue. Let me show you how that would look. So I am not a neuroscientist, so what the landmarks I will be clicking now won't make any sense. So there is this two-pane mode, which you can activate. I think this is always a bit easier for clicking landmarks. So if we zoom in now, I can now go to the landmark tool, which is this pin button. And then I click on the plus, and then I can set a landmark in the reference space. And then I can set the according anatomical landmark in the incoming volume, so in my own tissue. I just click something now, which doesn't make any sense, I think. And so you can click multiple landmarks. It makes sense to do at least 4, depending on the transformation type you are selecting. In the documentation, the differences between the transformation types are explained, so I won't go into detail on that because my landmarks are now not really reasonable. I just load some landmarks, which is also possible. So you can always save landmarks and then reload them later if you want to do that. And then you can click here on the compute button, and then it computes a transformation for you. And if we now go to the overlay mode again, we can then see how the alignment went. So this is not the original transformation, but as you can see, it looks quite nice. The main feature that I find very interesting is that you can now use this result in different ways. So, for example, you can either export your transformation and reuse it in other tools. For example, you can feed your parameters that you calculated into tools like ANTS, which can do a nonlinear registration. So this can be used as a starting point for your registration, for example. You can also create URLs, so you can share your data with colleagues, or you can also submit the volume as well as the transformation to EBRAINS curation, so we can make it available in the Zebra Explorer. But if you don't want to share and only take a look at your data in Zebra Explorer, this is also possible. I will show you how that works now. So if you go to this use result button, you can now click on the brain icon and then click on View Overlay. And then Zebra Explorer opens. Then you have to confirm that Zebra Explorer is allowed to load your data. Now let's pray that it works. Yes, it does. Very good. And now you can see your volume, the way you anchored it to the big brain in the Zebra Explorer. So you have everything available that Kim just showed. You can also adjust your appearance of your volume over here so that it makes it's a bit easier to see it. So also you can adjust opacity and stuff like that. And then here you can also, for example, switch the parcellations if you want to have a look at cytoarchitecture. Then it will load the parcellation. This may take a bit now. But then you can still see your volume. It takes a bit of time. Yeah, there it is. And then you can see the areas, for example, here, area STS1, that is next to your tissue. And then you can do the same stuff that Kim just showed, and you can explore how your volume is related to all the areas in the atlas. What I want to mention is that, of course, the big brain is an individual brain. Your data is usually also individual, so you cannot take it as 100% that this area is now intersecting with your tissue, because this is only a linear transformation, which doesn't put it exactly on the big brain. So it's also, yeah, it's still, yeah, very probable that this area might be there, but it's not 100%. That's what you should take into account when you're doing this. Okay, yeah, that's all from my side. What time is it? Okay, yeah, that's fine.
Finished.
Yeah. So thank you for your attention. I know it was a lot of information now from our side, but yeah, we also have documentation on all of our tools, so feel free to look there. And now, yeah, I give back to Sebastian about the hands-on part.
Yes. So normally, if we do such a session like this one, we do it in person. So therefore, we start the hands-on part and we walk between the tables and help the people. This won't work here in such a virtual situation. So therefore, we prepared some exercises. Those are just ideas for you to start playing with the viewer. Because it's always, if somebody says you please start playing with the website, do something, normally we have no idea what to do. So therefore, please don't see it as an exercise. It's really only ideas to get used to the handling of the Zebra Explorer. And we thought it would be best if we have about 15 minutes, something like this, time for you. Just go through the to the document, see if there are some of the examples which might be interesting for you. And at the end, so there are still 10-15 minutes left, we will go through the document together with you to show all the things we have asked for in the specific document. So I hope everybody of you was able to open the document with the exercises. Here now you can see the all those links we provided with this exercise sheet. So please take a copy on your PC. You will find all links to the Explorer, to the documentation, to the knowledge graph, and everything which is needed to solve those little questions here in the document. So we thought it might be best if we go through those first very basic questions here. I will go through the document and Kimberley will show it in parallel. So maybe Kim, you can share your screen and I will go through the exercise so that we can yeah, go on here. So the first one was very easy and basic question. How many atlases are available in the Zebra Explorer?
So just open Atlas here. You will see we have 4 different atlases here.
Okay, and the next one was find the claustrum in the right hemisphere of the monkey atlas.
So the easiest way is always just type in. Ach, we have monkey atlas.
Yeah.
I forgot to switch the brain. The left one, right one. With a 50-50 chance now. So here we see the claustrum in the monkey, in the macaque brain.
And you directly answered the second part of the question, what is the centroid of the specific structure? So you selected the area by using, you searched for the name, then you clicked on the centroid button from the feature panel, and then you can see the centroid displayed on the left side, and it's also written at the top of the screen. Yes. Okay. Next one. How many reference spaces are in the atlas, in the human atlas?
In the human atlas, we have the big brain, the FS average, the ICBM, and the MNI colon. So we have 4.
And what's the main difference between those reference spaces? So I can answer that well. So one of the most important differences is that big brain, ICBM, and MNI column are volumetric reference spaces, and the FS average is a surface-based reference space, which are 2 different type of representations. The big brain is a high-resolution reference space, whereas the colon 27 and MNI 152 are low-resolution 1 millimeter reference spaces. Okay, could you please select area FP1?
Mm-hmm. Now I do it by clicking. Something different now.
Okay, so after it's selected, you can directly see the centroid, which is the next part of the question. And you could also navigate to the centroid like Kimberley just did. And the second part of the question is, what would be the reference, the centroid in the different reference spaces? And this can either be done if you go to the reference space and search for the area specifically, but since we said that those spatial properties are reserved between the reference spaces, you can simply switch the reference space to MNI or to big brain and directly see the centroids in the different reference spaces, which are of course slightly different since they are different reference spaces. The next question, and this is from my point of view a quite important one, which area can be found at the coordinate 56, 12, 14 in the MNI space?
56.
12 and 14.
12, 14. I will just navigate to the point and have a look. We have here area 44.
Yes.
I'll just have a look at the list.
As I said, I think this is quite important from my point of view, because this is a typical question you have if you have your own volumes of interest, you have coordinates reported in different studies, so you can directly overlay them and assign them to different parcellation schemes here and directly see which structural area is located behind the coordinate you are looking for. Okay, the last point we will go through is the amount of different parcellations for the MNI colon space.
Well, the MNI colon space.
Doesn't matter.
Yeah, I could also stay here, but we have here 5 different parcellations with 3 to 5 further parcellations.
And the second question would be how many areas are included in the specific parcellation I was looking to. And this can be solved by clicking on the information icon next to the parcellation, because then you have some information about the amount of areas which are included, and also directly the link to the knowledge graph, which is also very important because there are a lot of data repositories out there in the web. But all those data sets which are available here in the Zebra Explorer are really deeply curated with a lot of meta information so that you really can work with them and include them in your own workflows. And the difference between the probability maps and the maximum probability map is that individual probability maps do show the probability of being a specific area in 10 brains, and the maximum probability map the label, which you can see on the 3D volume, which was calculated using a winner-takes-it-all model, and gives you also probability, but in a simplified way. So if you have the chance, use the probabilistic maps. But if you need whole brain parcellation, then we suggest to use the maximum probability map. Okay, and the rest of the exercises are more or less combinations of different questions and different tasks we have just shown here. And since we are running out of time, are there urgent questions? Is there something where we can help? And as Sanne already wrote in the chat, really please feel free to write us emails because it's still under development. So and at the end, we need feedback from users. And if something absolutely not intuitive for you to solve and you are completely lost, this is also important information. But of course, we're also open for positive feedback. So yeah, at the end, I would like to thank you for your attention. I hope you could give you a rather broad overview. And if there are no urgent questions, I would say that we are more or less done for this session. Tomorrow, Timo will continue and show you the Python part. So everything we have seen here can also directly be done in Python. And this is something Timo will show you tomorrow, also in an interactive way. So he will write some Python notebooks together with you so that you can really run them directly in the browser. And yeah, hope you have had a good time.
Thank you.
Thank you.
Goodbye, everybody.